Good afternoon, student, and I think things are going good in terms of our virtual classes. And I think I have already sent you the slides of mRNA processing, how the processing of mRNA takes place. So I think you have gone through, and even I also sent you the slides for same. So what I will do in this presentation, in case if you have missed something, so I will try to go through and make the things more easy to understand. So you can clearly see that the first slide is the mRNA processing, so I will try to skip. These are the certain slides, like the basic understanding of what is RNA, so I'm not going to go so long. So you know what is RNA, what is the primary structure of RNA, so we know these things. And these are the different types of RNA, like messenger RNA, ribosomal RNA, transfer RNA, and these are the function of these guys. So I'm not going to talk so much because we have already explained in this the virtual online class. So I will go quick to the next one. Similarly, like the ribosomal RNA, so these are the side for the protein synthesis. And yes, here. So if you like the structure of the gene, it consists of the protein, the coding sequence. You can see the yellow bar over here, and then there is a 5' UTR and the 3' UTR. So these UTR would be transcribed, but they are not translated. So these are the three main parts: the protein coding sequence, the 5' UTR, and the 3' UTR. Now the big question is there: why, where, and how the RNA processing goes on. So reason behind that, the RNA processing is important because the pre-mRNA need to process to become the mature RNA, and after that it can be easily transported from the nucleus to the cytoplasm for the translation. So the mRNA processing takes place in the cytoplasm, and once it is done, then the mature RNA move from the cytoplasm to the nucleus. So in a very simple way, it is nothing extraordinary. It is simply this: the pre-mRNA undergo certain alteration, or these mRNA undergo some molecular events, or finally these molecular events are like splicing, adenylation, and 5' capping it. When all these processes are done, then this mRNA is processed and it is ready for the protein synthesis. And these processes, if you go back to these, are carried by some nucleases. We know that these are the ribonucleases that can also create the RNA, and they can also take an important role in the turnover process. So again, I'm not going to talk turnover because we have already discussed. Turnover is simply means degradation of the RNA. So turnover, you should not confuse with the mRNA processing. So mRNA processing is not the degradation. It means the mRNA is processed in more stable RNA so that it should be ready for the protein synthesis. So if you look here, the green circle, there are the three important events: RAM, removal, addition, and modification. So these are just the three important processes that takes place in mRNA processing: removal of nucleotides, addition of nucleotides at the 5' end, 3' end, and then the modification of the certain nucleotide. So these processes takes place in the nucleus. Once these processes are done, then the primary transcript will change into the mature RNA, and this process will lead to the mRNA processing. And I can clearly explain here. So there are the 5' capping end, there is a 3' polyadenylation, and also there is addition of methyl group at the 5' end. So these are the three big events, or the main events that lead for the RNA processing. So remember: RAM, removal of nucleotides, addition of nucleotides, and the modification of nucleotides. And the whole process consists of the snRNP particles, and these are the guys that take part in the mRNA processing: hnRNP, snRNP, 5' capping, cleavage, adenylation, splicing, and the methylation. So let's go one by one. And again, these are the certain enzymes that are involved in this process, like RNA pol2 takes place in the pre-mRNA, then similarly the siRNA, the pol5, so you need to just go through it. One thing you need to remember: in prokaryotes, there is no processing of mRNA because there is no intron And moreover, there is no clear-cut compartmentation between the nucleus and the cytoplasm. So once the mRNA is transcribed, it has to be translated, otherwise it could be degraded. So in the case of the prokaryote, there could be no processing because they also contain no introns. And this is a simple process. So here it is DNA, the transcription. This is the pre-mRNA. Then it undergoes the RNA processing to become the mature RNA. And once this is done, then these guys move from nucleus exported to the cytoplasm for the protein synthesis. So the processing of mRNA is done by the RNA polymerases, and they can form the polymerases, the group of these RNA, and these are called as the heterogeneous nuclear RNA. And these mRNA give rise to the mature pre-mRNAs. And these pre-mRNAs undergo the capping, cleavage, adenylation. So there are one more slides, that is this, what is HNRNP? So HNRNP, it is the heterogeneous nuclear RNA plus protein. So it means the RNA which is formed by the RNA polymerase, if they are combined with protein, they can form the HNRNP, and this is very important in the mRNA processing because these guys will keep the pre-mRNA in a single strand and help and assist in the RNA processing reaction. Similarly, the snRNP involved in the process of spliceosome, and they are called as U1, U2, U3 because it's the uracil. And these are the three steps. So for the 5' capping end, these are the three steps. The first, there is a removal of the phosphate from here. Then there is the addition of guanosine nucleotide, and once this is added over here, then this guy will be methylated. So it is also called as a 7-methyl guanine. So this is the first step of the RNA processing, and that is also called as the 5' capping end. The next is the polyadenylation. Again, you need to remember two things. Here it is not simply the polyadenylation, it's the cleavage and polyadenylation. So there are the certain signal which is present in the pre-mRNA, and these are called as polyadenylation sequence or the signal. Once this signal is recognized by the endonuclease, and these signals are present after the stop codon because they can be transcribed. Once this signal is recognized, then the endonucleases will cleave roughly around 20 nucleotides downstream. Once it is cleaved, then there is a shorter, and then the poly-A will add the number of adenine nucleotide, and this will give to the polyadenylation. So it is simply the addition of adenine nucleotide at the 3' end, but before adenylation, these pre-mRNA need to cleavage at the polyadenylation sequence. Now the important process is splicing. As I told you in the class, it is again the removal of intron and the joining together of exon. So both processes lead to call as the splicing. It is not only the removal of intron, it is both removal as well as the joining of these exons. As we know that the introns are the non-coding sequence and the exons are the coding sequence. And one point you need to remember here, during the splicing there are the 3 points. One is the 5' splicing site, one is the 3' splicing site, and one is the branch site. So the 5' splicing site, there are the nucleotides which is present, that is called as GT at this point and AG at this point. So these are the point where the splicing should take place, and this is also called as GT AG rule. And there is also the branch point which is also the conserved sequence and adenine, and then there is a formation of the loop over here. So you can see here, if there is a splicing of pre-mRNA, so first this is exon 1, this is exon 2, this is the 5' splicing site, that is GUGT AG, this is the branch site. Then the U1 will clearly come and they can bind with one of this splicing site, then the U2 will bind to the branch site, and then the additional snRNP will come and start with cleaving at the splicing site and the 3' splicing site. Once this is done, so these splicing site, they can form the complex body that is called as the spliceosome. Once these are the cleaved, so they are cleaved from here, they are cleaved from here, and this loop-like structure is also called as intron loop or the lariat or the intron lariat that could be easily removed after that. And finally here, then once this is taken out from the exon, these introns will be degraded, and again the SNRP will be ready for the next process. And now you can clearly see, once these are done, then these two exons are joined together, and this is ready for the processing. There is one more term, alternative splicing. So in the case of alternative splicing, like in the splicing only the introns are spliced out. In the case of alternating splicing, sometimes there is a selective inclusion or exclusion of exons. So when we talk about exons splicing, this is called as alternative splicing. So you can see here, in the first case, it is a normal splicing. Once there is a formation of these, the mature RNA, there are the 3 exons. Again, there is a splicing, and now there is a exon 2 spliced out. So this is also called as alternative splicing. And this is play a very important role here, because now you can see here, this is this exon gene organization of tropomyosin, and these are, you can see these blocks are the different exons. In some cases, if this exon is removed, this guy can behave like a striated muscle. Then if this one is present but this one is removed, or there is a small removal within the exon point, they are the smooth muscles. So these are the alternative splicing. Moreover, in the case of the Drosophila, the sex determination is also based on alternative splicing. You can look here, there are 1, 2, 3, 4 introns on here, and 1, 2, 3 exons on here. Here, if exon number 2 will be spliced out, then there is a joining of only 1 and 2, and if this is the case, then it will lead to the formation of female Drosophila. If all the 3 exons are present, then it is the female Drosophila, and therefore this is very important, and this is called alternative splicing. Similarly, the same case here, you can see there is a heart muscle. There are 1, 2, 3, 5, 1, 2, 3, 4, 5 exons are there. These are the introns. If all the exons are intact, 1, 2, 3, 5, then it is the heart muscle RNA. So here the exon number 4 is spliced out. If the exon number 2 is spliced out, then it becomes the uterine muscle mRNA. So what does it mean? So it means the single transcript. Keep this in your head. The mRNA, single mRNA will be translated into different protein. I'm not talking about 2 mRNA will be translated into different protein. Single mRNA will be translated into 2 protein. It totally depends if there is a alternative splicing. And almost in the case of human genome, almost 60 to 65% of this the human genome organization undergo the alternative splicing. So you can see here, this is the kind of overview. So there is an exon, intron, adenylation, splicing, the processing. So all this process together is mRNA processing. To sum up here, you can see here, you can start it with 7,700 base pair of over albumin gene. Once it is processed, it is only 1,872 base pair, and this will be transported to the cytoplasm for the final synthetic product of this, the translation. So again, in a very simple way, so this mRNA processing involves the capping, splicing, polyadenylation, and this will give rise to the final product. And now this is the final slide. So you can see here, there is a DNA, there is an intron. So this will be transcribed. Once the transcription, there is a pre-mRNA. These pre-mRNA will go under the 5' capping at the 5' end, that is called as 7-methyl guanine. Once this is done, then there is a addition of poly A, and as I told you before, the poly A, there will be cleavage. Once this is done, then there is a splicing out. So splicing of the intron is done, and then there is a intron lariat will move out, and finally these the exon will be joined. And you can see this is the mature mRNA 5' cap and no intron, and there is a 3' polytail. So I try to summarize the mRNA processing, but again, just to sum up the story, these are not the complex process, these are the important process that makes the pre-RNA to form into the mature RNA, capping, adenylation, splicing, and once it is done, then it will move from cytoplasm to the nucleus for the final synthesis. So I hope this is clear. I will upload this slide on the YouTube, and I can also send you the link to just watch through. In the meantime, again, if you have any query, you are most welcome to ask. Again, So beat Corona, stay safe and healthy. Goodbye. Take care.